Hello, everyone, and welcome back to the Potavirus podcast. So we are here today. It's just me today with Dr. Marisa Azad. She is at the Ottawa Hospital in Canada. As you all know that I tell you all the time, I'm Canadian, so I like to make sure we have Canadian representation. And we met, I don't remember how, but I was put in touch with her. She is doing phage therapy for patients in Canada. She's one of the first, and that's what we're going to talk about today is kind of her experience. And as we've been doing in this sort of phage therapy series, figuring out from the clinicians, from the CEOs, from whoever is building in the phage space or trying to move things forward, what are they dealing with? What are the actual hurdles that are most important right now? And preventing them in the clinician's case, what's preventing you from being able to treat more patients, and what are your wishes? And then maybe the universe will hear them on this podcast and people will kind of take strides toward fixing those things or working together to fix them. So maybe I'll pass to you, Marisa, and just if you want to give us a little bit more of an intro about how you kind of got into phage therapy and about yourself in general, because you had also a life before phage. So yeah, welcome.
Thank you so much. It's great to be here. I love your podcast. Uh, so I guess I'll start a little bit about myself. And before I entered into the world of phage therapy, it was quite serendipitous, actually. I'd always been fascinated with phage therapy. My specialty is in orthopedic infectious diseases. So I did an advanced ortho ID fellowship at the Mayo Clinic, as well as a postdoctoral fellowship. I did my PhD in microbiology before that. And I sort of was thinking about phage therapy in the periphery until I encountered a patient in my PGI clinic who was septic from a particularly serious hip infection or a periprosthetic joint infection, aka PJI. And this poor patient, she had relapsed so many times with staph epidermidis in the hip. And by the time I had seen her for the first time in clinic, she was in a very rough spot. She had something called DRESS, or severe allergic reaction to two major antibiotic classes, glycopeptides, beta-lactams. So I was unable to treat her recurrence of the staph epi PGI with any of those therapeutic options. And she also had toxicities with one of the only antibiotics that we could use, daptomycin. And she was septic when I saw her in clinic, and she was older, she was frail, very, very bad situation for the patient. And I remember thinking, well, it's unacceptable to just tell this poor patient, there's nothing I can do to help you. And in the world of PJI, optimal treatment, we think, is to surgically cut out all of the infected hardware. But she had such extensive hardware. She had so many operations, approximately 20 total of the pelvis. So cutting out her entire pelvic area, that's not feasible. And it has such a high morbidity and mortality. So that's when I thought, okay, well, what about kind of pushing the boundaries a bit here and thinking about phage therapy. So that was my foray into the world of phage therapy. And my research team and I here, in collaboration with Cytophage Technologies and Dr. Brad Cook and Dr. Stephen Terrio, we embarked on this journey here in Canada. Our group was the first to embark on this phage journey for PJI treatment. And thankfully it worked. It was quite a process getting there in terms of regulatory hurdles, but we recently just published on this N of 1 phage trial, and we have successful outcome. This patient, thankfully, she has not had any relapses of infection. She has complete wound healing. There's no drainage from the incision sites. She's healthy. She has moved out of her home and she's living independently, and her mobility has improved. And further to that, biochemically, her inflammatory markers have completely normalized for the first time ever. So
Wow.
it's quite exciting, and my research team and I are using that N of 1 phage trial here in Canada as a platform for a multi-center phage trial, which I'm leading alongside Cytophage Technologies. So we're treating other patients,
Yep.
not only in orthopedic infectious diseases, but I'm also interested in using phage here in Canada to treat other implant-associated infections such as endovascular infections, graft infections, and even respiratory infections. So my team and I are working with respirologists as well to try and apply this technology to that area.
Wow. Amazing. Yes, and I remember seeing the story because that story came out all across Canadian mainstream news, and like it's, something has happened when you're getting your phage news from, I'm getting them from my grandma and my mom instead of like my phage community. So I was like, whoa, we hit a level. What is this? And yeah, that was extremely encouraging to see. I want to talk about because I've been confused about this for a while. When we think about N of 1 trials versus compassionate use, can you help us understand the difference? And why does Canada do the N of 1 trial? Because I feel like that's not really what's happening in the US. They're just calling it the emergency IND or compassionate use.
Yeah, that's an excellent question. So the regulatory side of this world, as you know, is a quagmire. But I have to say that Health Canada has been excellent. They have really streamlined this process for my research team and I. And, it was a journey for them too. So for us, there are two streams. So you can do the emergency access or compassionate use. But for us, because I wanted to do a multi-center trial and use this as a platform for that, I wanted to go through the N of 1 or OLIP route or single patient trial route. And I spoke to Health Canada about this when we started early on and we had our pre-CT meeting and I explained that to them and they were very helpful and they said, in this case, it would make sense to go more of a clinical trial route if you want to keep going with this instead of doing these, kind of one-offs here and there. So that was the rationale for us.
Okay. So you could have the option, but this is kind of the choice that based on the kind of paperwork and kind of like what you were establishing, it would set you up to do this for future patients with like presumably less work each patient. And
exactly.
Does there have to be consistency in how you treat the patients? How does that work?
So for us, we're setting up for more of an adaptive trial. So I have my protocol and we're adapting it to each patient. So in phage therapy, as you know, and especially in orthopedic infections, no two patients are the same. There's so many factors you have to take into account when you're trying to design a patient-specific protocol. So we kind of have that personalized medicine component of it. But as long as you have the skeleton protocol, you've shown that we know that phage is safe, but you have to kind of show the rest of the world or the regulators that it's safe and your research team knows what they're doing. There's that trust aspect and it has really streamlined the whole process.
Okay. Okay, awesome. So yeah, and it's very encouraging that Health Canada is supportive. And I feel like this has come up in a thread of some of the episodes you've recorded to date, is that it's like, is FDA or Health Canada or whatever equivalent like the bad guy? Are they restricting this? And I feel like even back in 2017, when I first heard the FDA talk at the Evergreen Phage conference, and they were like, please, we want to help you, like call us.
Yeah.
And it just is totally two different things you hear. And so it's good to kind of get that sense that it's not the regulator blocking you, but there is like a burden of proof kind of on you to show that you can make something safe and you know what you're doing and you have like thought this through and it's actually going to potentially benefit.
Yeah, it's such a good point, and actually, I've heard this from my collaborators across the country. It's really the local regulatory hurdles which are the biggest challenge. Health Canada, at least in my experience, they've been so up to date. They're well informed of this technology. A lot of these individuals, they're scientists, so they've been very helpful.
So the local regulators, is that like on the hospital level or who are the local regulators?
Yeah, it's and I've heard this from my colleagues across the country. So you kind of have two separate regulatory hurdles you have to jump through. There's your local hospital-based ethics approvals you need to get, and then in parallel, there's Health Canada. So yeah, and every institution has their own quirks, shall we say, when it comes to regulatory challenges. So, you kind of have to really be well versed in that, especially when you're working with multiple centers at once. But we've gotten there in the end.
Okay. Yeah. And I guess I just want to touch back on the original patient case. And so how did you, I think that you said Cytophage was the one who actually prepared the phage for you, right?
Yeah. So Cytophage has been excellent. I know you know Brad. He's amazing.
I love them. Yes.
Really love working with Cytophage. They're excellent scientists. But not only that, they're just fun to work with, which is just as important, right? And so yeah, my lab collaborated with their organization, their company, to prepare the specific phage for a patient. Initially, we were thinking of a cocktail. We then kind of narrowed it down to a monophage preparation. I mean, that's a whole discussion in and of itself, cocktail
Mm.
versus monophage preparations. But yeah, uh, it's yeah, that's how we got our phage for our patient.
Okay, so you already kind of knew of them, or did you kind of have to cold email them and say, like, I have a patient, do you have a phage?
So when I started my phage journey, I spoke to my colleagues at Mayo Clinic, Dr. Robin Patel, who's my old mentor. She's fantastic. Dr. Gina Suh, who's amazing, and she's on her paper as a co-author. So I initially reached out to them, and I literally Googled phage therapy companies. And I,
Mm.
I reached out to multiple companies around the world, and it was Cytophage that
Wow.
pulled through. And I remember I met with Dr. Cook and Dr. Terrio, and I get just clicked right away.
Yep.
And I explained the situation to them. And the thing about their group is that they really cared about patients, and that was important for me. And it was just worked.
Yeah. And was that the first case they were part of as well? Like part of preparing for like a
Yep.
patient case? Yep.
Yep.
Yeah. Because they were working on the agricultural side, I think, for the first chunk of their time
Yeah,
in the
that's a
industry.
whole other cool area that
Yeah.
they're working on, yeah.
Yeah. Okay. So they agreed to make phage for you. So then you would have had to send them an isolate, and were they the ones that found the phage as well? They just happened to have a staff epi phage on hand?
So, our phage, we put out, like, a world call, like a phage call. So I've learned a lot about how this stuff works. So our phage initially came from Slovenia.
Ah.
So it was pretty cool to have this network where you can kind of say, look, I have this organism for this infection. Does someone have a phage that could potentially work?
You must have used the Phage alert then, our Phage
Yeah,
directory system.
exactly,
I'm
exactly.
like, wait a minute. I do remember a
This
staff epi
sounds
case,
familiar.
but like I didn't know you then.
It's a
That's
small
so
world.
funny. Okay. I'm like, yeah, I think there's not any other global alert systems for phage finding, but if there
Yeah.
is, great. Awesome. Okay. And then so you found it in Slovenia, and then that was shipped to the folks at Cytophage in Winnipeg at some point, and then they
Yep.
produced it for you.
Yep.
Okay. And then how did you decide how to administer it, and how did you administer it?
Oh, that is such a good question. We're still in the wild, wild west, right? This is like HIV in the 80s and 90s. There's a lot we do not know about phage therapy and clinical applications thereof. So I looked at what little literature we have for the treatment of orthopedic infections. And in Canada, no one's done this before. We used the protocol that we adopted to our patient. Um, there's some case reports of just giving one single dose of phage for PGI, and that's it. But if I wish I could show you an X-ray of her pelvis. The burden of infection is so immense. I wanted to prolong her intra-articular delivery of phage and give adjunctive intravenous phage as well. So that's what we did. We worked closely with interventional radiology here, and our colleagues there introduced a catheter into the predominantly infected hip. And I injected phage twice a day into the hip. We wanted to go for about 2 weeks. You have to be very careful because anytime you introduce a foreign body, especially a catheter, there's always a risk that you can introduce other organisms into the
Mhm.
infected space. So, I watched her like a hawk. My team and I watched her like a hawk. The nursing team here was great. So I was a bit nervous in the beginning when I was injecting the phage therapy. What's very interesting though is that my team and I noticed that she had quite an immune response, predominantly when we injected phage intraarticularly versus systemically. And I would have thought it would have been the opposite. And the patient even commented on it. She had some rigors, chills during early administration phases, a bit of hypertension, interestingly, and then she kind of dipped and rebounded to hypotension. It wasn't a true anaphylactic response.
Mhm.
And thankfully, she did very well, and these immune responses went down with each dose, and we had a
Mhm.
nice curve. I'm getting into the weeds here.
Yeah,
It's just
that's
something
great.
interesting that no one's really reported on before.
Yeah, I think that area is of huge interest to the phage field, and we're starting to like triangulate on like what's missing to be done. And one of the big categories I think is the immune response. And
Oh yeah,
I think there's been a lot of hand waving for a long time on like, well, there's no immune response. They're silent. They're in the body. And
yeah,
then there's like, oh, well, obviously there must be neutralizing antibodies. So
yeah.
you can't redose. And somewhere in the middle is probably the truth. Like probably it's phage by phage. It's probably different. And I know in Australia, when we were treating patients with phages, we started to collect that data. The serum samples that we were collecting and storing in the freezer right before I left, had a student in Bosco that was her PhD project, to start looking at all of those and see if there was antibody responses at least in those patients. And I'm pretty sure that it was very heterogeneous. Like one patient would, another wouldn't, and a
We
little
need
bit
way
phage
more data.
specific.
Yeah, yeah.
Yeah. So Kieran, if you're listening, tell us how your research is going.
Well, that's interesting. Another arm of my research is looking at the pharmacokinetics of phage. I mean, that also is a black box, right? How often do we dose? What dose? How do we deliver it? I mean, looking at things like hydrogels, there's so
Yes.
much we need to look into here.
Yes, yeah. Now you're talking the language of the lab I just joined. It's like pharmacokinetics and hydrogels, but
A nice
it's all
steady release.
delivery. Yeah, yeah. And like what will help and what do we need? Like what's a good profile for a phage? Like how long should it last?
Yeah.
And like should we deliver it more slowly, or and what's the dose? I feel like yeah, that's where the field is getting, but it feels like movement from like 8 years ago when it was feeling more like, okay, let's figure out what all the genes do.
Yeah.
I don't know. I don't
Baby
know.
steps.
Yeah. Yes. So I want to ask, how has your approach changed from the first patient that you treated to the ones that you're thinking about treating now, or the other ones that you have already treated? Have you learned anything that you're not doing anymore, or that you should have done the first time that you're now planning to do?
Yeah, I think I'm always wary to leave a drain in situ for more than a couple days. So, especially if it's the pelvic region, they are very prone to falling out just in general because you're moving around, and plus it's a very dirty area in terms of contamination, especially in older patients, and they can inadvertently roll and pull it out, and do you reinsert it? Do you leave it? So for me, I think for our next batch of patients, we're going to reduce that intra-articular dose duration. And then in terms of what we're going to do next, we're doing pharmacokinetics. That's something I'm really interested in. And then, things that I've learned from that first single patient trial. Really, it's the regulatory stuff and just little things like admitting the patient and ensuring that they have safe monitoring and getting everyone on board, sending out a memo to the nursing team. Little things that you don't think about until you actually go through it. Um, I'm really good at that stuff now too.
Yes. Yeah. I feel like there's so many unwritten things, yeah, that I learned in Australia too, like how to communicate with the hospital micro lab, like when they
Yes.
get a new strain, like they were just writing like phage in block letters in a Sharpie on the bag, and like, there was no way to put in their system like that it was going to our lab next if
Yeah.
it was
Yeah.
kind of resistant. And we were like, okay, we just would show up and find their office and be like, me and Stephanie, we're the two postdocs who are going to do this. Like, can we come up with a code that you write on this that like connects to the bug? And they were
Yeah.
like, oh, yeah.
That's a good idea.
Yeah, people were amenable, but it was like hard to find the right person the first couple times. And
Yeah.
yes,
It's the administrative stuff that gets you.
yeah,
My
yes. Okay. So you're shifting your approach, but you're still liking the intravenous route as kind of like the
Yeah.
backstop to whatever you're doing.
Yeah, and the next set I where timing is important. That's something else I really learned about. So because we can't get phage therapy overnight yet, we have streamlined the process drastically, and Health Canada has been very helpful, and they know that patient lives are on the line, but it still doesn't happen overnight. The paperwork in and of itself, I think I aged like 20 years that first, but the paperwork itself, my poor research team. But we have all of it done now, and it's just getting the timing right. So, because especially with orthopedic infections, things change so drastically. I tell my patients, do not stop your antibiotics until, X, Y, and Z, or, things like that. But sometimes I've seen funky stuff where patients will get like a viral URTI and then they flare despite being on suppressive therapy. Like there's not really any data in that, but that's something that I've seen in my practice, which is interesting. And they unexpectedly flare, and then suddenly we have a new organism, and the months that we took targeting that specific
Yep.
organism is now moot because now we
Yep.
have a polymicrobial infection. Or, there's a case we recently were working on with our colleagues in Concordia where the patient unfortunately developed an ascending UTI and then became polymicrobial. So, there are things that we cannot control. Bacteria don't care. They just want to make their home, make their little microbial niche and thrive. Things like that. I've gotten better at timing and predicting when I think someone's going to fail or
Okay.
flare.
Yep.
That's kind of a gestalt thing.
Okay. Yeah, I feel like our episode with Gina got into a little bit of this. Like she was saying, one of the things she learned was how to better pick who was going to be able to benefit, given like these unique constraints and strange wizardry of timing and like of knowing the pathogen. And I feel like my Australian experience, to go back to that again, like I was listening to the clinicians make their decisions, and it was interesting to hear them go back and forth debating whether like a given pathogen was likely to be the causative pathogen, because you kind of have to make a guess on that, it sounds like, based on what you've
Sometimes.
seen, and like if there's multiple things there, but one of them's, typical, yeah,
Or
like
like the
something in the
dominant
skin.
pathogen.
Yeah. And like we talked last couple weeks back with the Biomics CEO, Jonathan Solomon, and he was saying like the whole head of the snake theory with staph and diabetic foot. He
Yeah.
was really excited to try to prove out, and they feel like they have seen like that getting rid of just staph seemed to help the entire infection. But that was a bit unknown. It still probably counts as unknown still, but it did seem to work in their trial. And so, guessing is probably not called guessing in your field. You capital letter G, like uneducated decision.
No, it's so true. Nothing in science or medicine is ever 100% or ever absolute. It's based on probabilities. I I can't really explain it, but with orthopedic infections, it just makes sense. For enterococci, for example, it tends to be eradicated a bit more easily than other organisms. If you have polymicrobial infections, depending on whether you do an anterior approach or posterior approach of the hip, there's so many factors that go into it, and you see these like interesting microbial dynamics where they're trying to outcompete with one another, and sometimes there's cooperation, and then eventually you get this dominant pathogen, Pseudomonas or Staph aureus. But it depends on the host, it depends on the mechanism of infection, the pathophysiology, what immunosuppressants they're on, what surgeries they've had, blah blah blah. But
Yeah.
it's a bit of an art form.
Yeah. I also didn't realize you had also a PhD in microbiology before you went down the whole medicine route. That's that's perfect. Are
Long
you
haul, but
tired?
worth it. Yeah, I did a combined MD PhD at
Okay.
McMaster University. It was
Wow.
a lot of fun. But
Yep.
when my friends were studying for exams or relaxing at the bar, I would be in the lab slaving away with my enzymes, my acetyltransferases. Yeah.
Yes, yes. Well, it's think it like is culminating so well, and it's so rare that you can have one person with both sides in one, like the clinical and the microbiology, the deep microbiology experience. And
Yeah, it's as you know, it's important to have, and it's fun to apply it to medicine, and it just gives you a different lens to approach infections by. I tell my patients and my residents and my learners, well, bacteria are like humans. There's bacterial ecology, there's microbial ecology. They're just trying to survive like the rest of us.
yeah.
Anyway.
Yeah. Yeah, that's awesome. So I guess I want to go to the next couple of questions that we tend to ask people. So we'll eventually put out some comparison of everyone's answers, I think. But what is it that you need the most at this point? Or what, if you had a magic wand to wave, like what's the challenge that you just wish you could just get past at this point?
I think, that's a very interesting question. I think there needs to be, well, okay, number one, regulatory hurdles. They need to be streamlined more, but we're getting there with Health Canada. Honestly, I can't say enough positive things about them, but I think locally, that needs to get up to speed. So there's a lot of misinformation about phage. It's very safe. There's so much data on its safety. That's one
Yeah.
thing that we have, right?
Yeah.
So I think that kind of needs to be far more supportive of experimental therapies, not just phage therapy, but
Yeah.
CRISPR and all of this
Yeah.
other cool emerging technology that can literally save lives or potentially save lives. We need support to do that. Number two, this maybe I don't know if anyone else has said this, but we need more collaboration. Some of my colleagues who've been working in infectious diseases for 40 years, they've commented that the phage world, it's kind of like I said before, like HIV in the 80s and 90s, it's a wild, wild west. And it's really important that people work together and something as simple as making phage libraries. I know multiple labs that are making their own phage libraries. Why don't we
Mm-hmm.
just have a central giant repository where everyone feeds all of this data into one site? We all use it, we all work together. That's a very kumbaya, let's all hold hands. I mean, science can be very cutthroat, unfortunately. That's just the world that we live in. With any new emerging technologies, it's important to focus on patient care. It's important to focus on why we do research. I think it's really fascinating. I'm an artist as well, not to get off on a tangent here,
Yeah.
but art, science, and medicine, these things all go together, and you got to be creative with it. You got to have fun with it and just work together. As cheesy
I
as
love
that
it.
sounds, I think that is how you really push things forward
Yeah.
together, like as a whole, and this isn't just for phage therapy, it's for science in general. All
Yeah.
right, I went on a little diatribe
I
there.
like it. Yeah. Well, I've been thinking about art and science lately too, because I think there's been this separation of the two for so long, and at least when you talk about careers and are you going to art or science, and science is the opposite of art. That's what makes it science. And I have been talking to some of my students lately and just saying I actually think you should see a career in science as a bit like a career in art,
Mm,
because it's like you kind of get this. It's hard to get um, yourself set up to do it, but if you can get a source of funding so that you can creatively kind of play in the space and figure out what works and what doesn't. It's kind of like these patronage models of a long time ago, and how science started too. You would, if somebody would support you to paint or to create, same with the science, it was just people who had that setup, and it wasn't for everyone, and it's not a right, and it's not really a direct function for society right now. We must discover this thing today. It's it should be considered more like a bit of an art or setup, like a patronage model. And that's what we're all doing. When we're working in a lab, we have gotten this space and a bit of funding but then we're allowed to be completely creative, and we should support that in that category. And yeah, and acknowledge that, yeah, it's not lucrative and super function driven. And then because of that, that's when such cool stuff can spawn out of it. But if
yeah,
you kind of try to make science feel more like what it isn't, where it's it has to be done this way and one step and then the next, and everything has to map back to use today, then if you did that to art, which has happened, of course, with the commercialization of it on one stream, if
yeah,
you did
mass
that,
production, yeah,
yeah, you kind of ruin it, but you can also have that channel, and that's great. And I don't think you ruin it. I'm not making a moral judgment that you ruin it, but the thing that you're doing, the creative, open-ended version of that can't it kind of suffocates in that model. And so I think we should just see science like that, and also medicine, with this experimental stuff, it is this creative sheltered little space where you kind of get some leeway from the regulator to be hey, nothing works. why don't you come up with something? We will support you and we'll go slow and like make sure it's safe. But we should all like see the privilege that it is to kind of get to be creative and I don't know, like, yeah, the working together and being like really open-minded about how to do it in like these like special little spaces where we're not behold to these, I don't know, like that are the way it should be done. I don't know. That's my
I love that.
rant.
Yeah, no, let it out. No,
But
that's
yeah,
great. That's great. And, I agree. And science, like art, it should be free flowing.
yeah,
We have the never ending, hamster wheel of getting grants and publishing, but you can't forget why you went into science in the first
yeah,
place. And I love that parallel you drew between the tortured artist and the tortured scientist. I mean,
yeah,
you got to suffer a little bit,
yeah,
a little bit, not too much. But, it's anyway, I wish there are a lot of we're getting way off topic, but
totally. That's okay. I'll allow it.
yeah, I mean, academia and there's a lot of things that need to change, I think, to promote that creativity and to promote that collaboration. But that's a whole other discussion.
Yep. Yeah, totally. Yeah. I've been thinking so much about how like compassionate use and N of one phage therapy and just everything with phage therapy fits into the world. And this is like the best place I see it instead of like shutting it down and saying, oh, it doesn't fit the standard mold very easily. It's personalized. So therefore we shouldn't do it. And it's like,
Wait,
yeah, I think it's like a hugely interesting space and if it did fit the paradigm better, it would be it would be hard to innovate in that space and it wouldn't be so open to creativity. So
yeah,
maybe it's like drawing us in and you're an actual artist on the side. I'm not. I just,
everyone can be an artist. Don't be so hard on yourself.
yeah, as a podcast art, yes.
Yeah, there you go.
Yeah. Okay, so more collaboration and more working together and I think, yeah, creative thought. I like that. So for the future, where do you want to be in 5 years with what you're doing? And what do you see? Maybe where
Ooh,
you want and what you actually think will happen.
ooh.
Pick pick one, pick both.
Very much for just kinda living in the moment, not to sound like
Yep.
a hippie, but at the same but at the same time, I really do think about the future a lot in terms of what's next. I like to build things. I want to make this more readily available to people. Just like you said, oh, personalized medicine, when there's a will, there's a way. And my team and I were already beginning to show that we can create an adaptive protocol and we can make this faster. And I think in 5 years, my team and I, and people like you and our colleagues at Mayo, we're going to keep pushing this technology forward, making it more readily available to those who need it the most, and answering a lot of those empty tick boxes for optimizing this technology for not only orthopedic infections, but a variety of other infections that are often chronic, recalcitrant, and debilitating. So for me, I just want to keep doing what I love. And I don't know exactly what that's going to look like, but we're building a network around the world and working with, good people like you. Yeah, I don't really know if I answered your question, but maybe just keep doing what we're doing, and I know that we're gonna be successful in some way. I just that's just my personality. When I get obsessed over something or really
Yep.
focused on something,
Yep.
everything kind of just fades away in the background, and I just have to do it. And for me, I this is just something I never thought I would end up doing. uh, Another tangent is that I never thought I would go into medicine, but it just kind of like things just kind of present themselves and you just kind of go with the flow. And my personality is I like to push it and see how far I can progress this area.
Yeah. Yeah, I love it. Yeah. I'm similar. Yeah.
I
It's
can
like,
tell. Yeah.
when people are like, what's your 5-year plan? Like I have this question and I'm like wincing as I ask it. We're like, oh, I hate this question because I would much rather just run at something
Yeah.
until I decide that it feels like enough. And I feel like sometimes putting a plan together, it limits more than it expands. Maybe I don't make big enough goals or something,
No,
but
no,
like,
no, no, I completely
yeah,
agree. It limits
yeah,
you.
yeah. And it makes it hard to run at it because I'm like, well, I've already imagined the outcome.
Yeah,
And like, I would rather leave that open ended and be like, imagine the outcome, but I'm not actually imagining it. Like
yeah,
it's bigger than my imagination and that's what's exciting, but we
yeah.
can kind of, yeah.
But
Yeah.
you
So
know what? You know
yeah,
what?
go ahead.
But, you talking about that, I can see how passionate you are, and another thing that I really want to do is support other women in research. I think that is critical. This job is hard in and of itself. Women have to work twice as hard as our male colleagues in many aspects of academia, science, and medicine. Again, it comes back to that collaborative, supportive spirit. You can be hyper focused and the best scientist in the world, but if you are not particularly kind or supportive of women in science, I mean, we this is like it's 2025, and that also is very important to me. And that just hearing how passionate you were there just really kind of reminded me of that goal or that part of my life as well. That's important.
Yes. I love it. Yeah. Yeah. It is. It's still an issue. Yeah.
That's a that's a whole other podcast discussion too.
Yes, we've covered phage therapy, art, science,
Many
women in
grounds.
science. But yeah, it's kind of all tied together. And it feels like any one of those things can be like at the forefront of your daily challenges. And it just kind of like isn't one. Like, I wish I could sometimes just, yeah, work on like one thing and not be like life stuff. Like, I'm planning my wedding right now. And it's
You
like,
are?
yes. And so
Congratulations.
my whiteboard, thank you. My whiteboard has all this like flowers related stuff. And then like also like figure out if we can deliver genes with phages and just like I feel like it's not talked about enough how
I
like,
love that though, that
yeah,
duality.
it's on one whiteboard because like I can only have one list, but it's kind of like what, like everyone's list must be so heterogeneous in their own ways, but I feel like we represent ourselves as one facet in any given context, and so it's like that's for another, I talk to my friends about the wedding stuff and like my
Yeah,
scientist people about the
you gotta
science,
yeah.
but
Yeah,
it's not really my like real life, I guess. It's like only one piece. So
exactly. We have all these facets to us and we're juggling all these balls and,
yeah.
it's a lot of work. But
Yeah.
congratulations, that's very
Thank
exciting.
you. Yes. Okay. So moving on to my questions, and we will tie this up soon. Yeah. So I guess if you had to say what research is actually left, and I know that you talked about the pharmacokinetics and the immunology, but since I think a lot of our listeners are phage researchers, what would you say is actually like a burning area that they should be working on if you were the one that got to decide? Like, are there areas that people, it's actually a solved problem from your view as a clinician? We don't need to study anymore. We need to study this. How do you think about that?
I think that we really need randomized control trials to answer a lot of these big clinical unknowns for phage therapy. But getting there is hard, and you have to actually know what you're doing. With PGI, orthopedic infections, it's such a heterogeneous patient cohort, difficult to design trials just because of that. But I really think, like, I have so many questions to ask, both as a microbiologist and as a clinician about phage therapy. Something as simple as what infectious diseases should we be applying this technology to improve patient outcome? And how do we do this? How do we deliver it? We've already talked about things like dosage, monophage versus cocktail preparations, infusion rates. Like, there's so many things we don't know. I think really we just need big trials once we kind of answer the little questions. So for the pulmonary cases that I'm looking at now with my team, do we do like there's nebulization? I mean, that's a whole other set of research that we could be looking at. And then you have to take into account pulmonary anatomy. Do they have stents? How severe is their disease? Can the phage actually penetrate these aspects of the lung? There's so much we don't know. I can't really answer that question because I would just rant for about an hour.
Yeah. 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 I wonder, like, sometimes I think about whether sometimes I think phage therapy, we know enough to like move forward, like, and yet on the other hand, yeah, you can rattle off like a million things we don't know. And I try to put them in categories of like, well, if we knew this thing, would that like change what we're doing or like stop us or help us, and mapping things back to that. Like, has there been something that you've found, you read a paper or you talked to somebody and you like use that in your practice, or like, I don't know, how do you rank things like that?
Like practice changing
Yeah, or like what's
things.
just like an example of something that actually like do our research papers matter?
That is another great question. So I remember when I was doing my PhD, I love biochemistry, I love microbiology. I would focus on this tiny subunit of this
Yeah.
resistance enzyme.
Yep.
And I remember when I was very young and naive, I was like, you know what? This paper is going to be huge. It's going to affect so many patient lives. But then I went back into medicine and I'm like, wait a minute, there's such a disconnect between the basic science and the patient. And that's one of the reasons why I wanted to be a clinician scientist, is to bridge these two worlds. And, something that I'm also really interested in is biotechnology, bioengineering new
Hmm.
implants that can prevent biofilm formation from happening in the first place, which is that at the crux of not only PGI, but so many other implant associated infections, even non implant associated infections. Like, you can get biofilms forming in solution. You don't even need an artificial surface.
Wow.
So I'm getting off on another tangent, but yeah, there's such a disconnect between what we do in the lab and what the patient has to suffer with. And I really want to make my research not only translational, but to improve outcomes for patients. It's very generalized, but anytime I do a paper or like a study in the lab, I always kind of think of that in the back of my mind because it's such an important point.
Yep. Yeah. And it's hard because acetyltransferases and patient experience. Like I studied one protein in my PhD. It was, glycosylation and a phage protein that seemed to, maybe bind to one of these glycosylation proteins. And like how, like that was, a point in my PhD where I was really depressed about everything because I was like, wait, this is never going to help anyone. But some people do a really good job of like keeping the two things, the two scales of like
Yeah, they're both
connecting the two
important.
scales.
And it's fascinating talking to like a pure scientist versus a pure clinician versus both, because they look at the world differently. They approach problems differently, and I try to learn from all three of these individuals, and it affects how I practice medicine. And um, it's these little discoveries in the lab that
Hmm.
can lead to these big randomized control trials and can lead to a new drug 20 to 50 years later,
Yeah.
if you're lucky. But there's always that hope and that trajectory. And what is that saying? We stand on the shoulders of giants. That's what science and medicine is. And it all comes down to working together and building off of our colleagues' work. And and that when I was doing my PhD, I didn't really kind of absorb until I went fully into medicine again, was that, bacteria, proteins, molecules, they behave very differently in a little Eppendorf tube or a Petri plate or a rat versus or a poor little monkey versus a human being versus a population of human beings.
Yeah.
So there's just that huge progression that we as researchers have to keep in mind as well. So sometimes I'll look at these really fascinating basic science papers, massive impact factor, nature, science, super sensationalized. But I sit back and, the PhD side of me goes, oh, that's awesome. But then the medicine side goes, okay, well, but I know that we're a long ways away from this positively impacting patients. And we don't even know if this is going to work in a human being because it's just a whole different ball game. You have your ADME, your PKs, you have your, stability of the molecule. Do you co-package it? How do you deliver it? The drug side of it, the manufacturing side of it, I've learned a lot about with these phage studies we're doing as well. So that's a whole different discussion too.
Yeah. Yeah. How do you not go insane? Like, how do you balance? Because you run a lab and you treat patients. Like, what is that? how much brain space are you switching between the scales? Are you trying to make them coalesce so that it's less, crazy making? Or what does it feel like to do both of those things?
Yeah, it's not easy. There I get bored very easily, and I think you're similar, like, there's just
Yeah.
always something going on in the background, like a million tabs are open, and, they're in my mind, it's just one entity, but I do need to kinda switch gears a little bit, between seeing patients, it's seeing patients, there's that whole emotional aspect of it that doctors tend to forget about over
Yeah.
the years. This is a human being, you're not just throwing drug X or Y or Z or, doing surgery ABC. The human being, they're suffering greatly and they are looking to you for help. That is important to me, so I put that side of it. But then with every single patient encounter, like I saw a patient this morning with unfortunately a missed knee infection for at least a year, and she was pretty sick. But I just explained to her the microbiology of it. So I brought in that bench
Mhm.
science.
Mhm.
And she said, "Wow, I didn't know that these little critters are acting like this
Yeah.
and behaving like this," and
Yeah.
it helps them. So I kind of like to marry the two worlds together every
Yeah.
day.
Yeah. Wow. And is your lab like near where you treat patients? Is it like connected to the hospital?
Yep, it's in the cancer
Yeah.
center.
Cool.
Short walk.
Wow. Yes. I've been to Ottawa a couple times and it's a nice place. And I did 5 weeks of a research sort of like visit at the
Cool.
it would have been in the School of Medicine, so maybe that's near where you are.
Yeah.
And yeah, anyway, another tangent, but good place. And it was, I always like joke that all my PhD work, all the papers came from like that 5 week stint in Ottawa. And so
Just
it's like a
bang 'em
special
out.
place. Yeah, it was late in the degree, things needed to happen, and I generated a bunch of data and like, I don't know, because of some awesome collaborators, they'll let me come hang out with them for 5 weeks. So
Awesome.
a nice supportive environment over there.
Yeah, we'll have to come visit again sometime.
Yes, I know. I haven't been back in a while. And you should come visit us down at Stanford if you
I
want.
would love to.
Yes, we're doing all kinds of PK studies. We're trying to get
Awesome.
grants for more of them, and we're still working with GINASA. And so we're trying to, yeah, like get grants for clinical trials. I feel like that's like the next frontier. And I feel like, yeah, companies are starting to do more, but you kind of have to have the investigators
Yep.
at all these places like kind of also push them forward. So I think there'll be lots of room to work together, and hopefully the shifts in government are going to have something positive for this field. I don't know. Like there's nobody's saying
Well,
anything positive, but I have hope that any shakeup on the regulatory side, there could be some openings that we could kind of find that could
yep.
help. Like
Yep.
maybe it'll be easier to do phage therapy in a sense if there's some pathway that, people have the right to get more options. I don't know.
Yeah, more
So fingers
alternative.
crossed.
Yep.
Yeah. Yeah. And I think we should see this as like an interesting turning point and not be only
Yes.
sad.
It's an interesting time in the world. We'll leave it at that.
Yes. Yes, exactly. And in Canada too. It's all
Yes.
shifting at once. It's all connected.
Changes.
Yes. Well, thank you so much, Marisa. This has been very delightful. And I like that we took this a few different directions than my standard questions. I don't know. Yeah, the human side is what's like, I mean, we're human beings driving all of this. So
Yeah.
it has to be
Yep.
part of it. And it's hard to find space for those conversations as well. So
Yeah,
we will
absolutely. Yeah.
make it happen.
It was great talking to you. I'm glad we connected, and let's chat again soon.
Yeah, I'm excited to keep up with what you're doing, and send me all your news and we'll just blast it to the phage community. And yeah, I'm just so happy that you're doing this in Canada.
Thank
Yeah,
you.
it's awesome. Okay, great. Thank you.
Thank you. Bye.
Bye.